come in next time. Chapter 8 is all about genetics. So again, the themes are going to roll over from Chapter 7 because 7 was giving you the background for this. This historical glimpse is pretty interesting. We've talked about Barbara McClintock a bit in the past. Remember, she's the one that looked at that Indian corn, the stuff that you're going to see around coming in this month, those cobs of corn that some of the kernels are kind of blue or black, and some of them are pink, and some of them are orange and yellow. And she asked the curious question, how do they get that way? Why are that way? And she was a geneticist, and so she started doing some genetics. She would take a couple of purple kernels, plant them, grow plants up, and then cross them. And according to Mendelian genetics, you would predict that all of the kernels, if you did that enough, crossing purple kernel plants, pretty soon you would get a plant that had all purple kernels. But what she saw was that sometimes she would get plants that had purple kernels, but then all of a sudden in the middle of a cob, she would start to see kernels that were yellow and pink and other colors again. And she started to form this hypothesis that genetics aren't quite as stable as what Mendel and other scientists had said. The only problem, well, there were two problems. This was back in the 1940s, 1950s, a little bit later on. She was doing all this research, and basically she was one of the few women in science. That was the first problem. The second problem was she was seeing something that was totally unheard of and very against the dogma of science, that genetics, the genes on chromosomes are stable. They're stably inherited. You marry somebody with blonde hair, blue eyes, and you have blonde hair, blue eyes. Theoretically, most all of your children are going to have blonde hair, blue eyes, and we could predict based on genetics what's going to go on. But she was seeing something that was very non-predicted based on Mendelian genetics and how genetic segregation is known to work. And so she published a lot of her results, but almost everyone that heard her results were skeptical. She tried to publish them over and over again, and people pretty much laughed at her. Pretty soon they wouldn't let her publish. Pretty soon they wouldn't let her present, and she just kind of gave up. And for about 10 years, she just continued to do her research and find more evidence to support the conclusion that she had come to on this in this particular organism. And then something curious happened. In the 1970s, two guys came along and they started looking at bacteria. And in bacteria, they saw something that was very curious. Some genes that they were looking at would be turned on, they would be working, and then a few generations later, they would be turned off and they wouldn't work anymore. But curiously, if they grew those cells where the genes were turned off, they were mutants, they grew them for a little while longer, the genes reactivated, and now the genes were functional again. And they started seeing this phenomenon, and they said, this is really curious. This looks a lot like what Barbara was seeing. And Barbara identified these pieces of DNA that jump around in the chromosome. They can be in one place in the chromosome, and the next generation, they can move nicely and cleanly from there, and they'll move over into another place in the chromosome. And some of them, there's lots of different ones that are known now, will move from one place, and when they move, they actually move and convert into 3 or 4 or 5 different copies that scatter throughout the chromosome. And so since then, she got the Nobel Prize in '83, and since then this term called transposons has come about, and we'll talk a lot about those in this process. But basically, they're genes that appear to jump in the chromosome, and some of them actually do move in the chromosome. So little bits of DNA that move from one place to the other. The curious part is, when some of these move, they jump in the middle of a gene. In the case of Barbara's situation, they were jumping into the middle of the gene that makes the pigment that turns those kernels the dark blue color. And then when the transposon's in the middle of that gene, they can't make the pigment anymore. They turn to one of those shades of pigment. But later, they can precisely jump back out again. In other words, the DNA is cut very precisely, so not one extra nucleotide is nibbled from here, and not one extra nucleotide is nibbled from here, and that transposon that's in the middle is chopped out, and then these ends are brought back together and glued back together, and now the gene works again. And now they can make that pigment again. And so we've seen transposons in a lot of different places. The organism that I did my PhD on, our lab, we identified one transposon. It's really cool. It jumps whenever you convince, it's kind of like an STD. Whenever you convince the bacteria that has these transposons to have sex, the thing jumps all over the place. and it gets really excited whenever the bacteria has sex. I don't know why yet, but it's a pretty interesting picture. It only really moves when the bacteria is mating, but when it goes to mate, it moves all over the place and disrupts all kinds of genes, and it's a pretty cool phenomenon. So it's a STD for a fungus, if you will. So back to this chapter, there's a lot of interesting things that happen, and we're going to talk about genetics in a lot of different ways, but one thing that we're all concerned about is antibiotic resistance. It's becoming more and more in the media. Organisms are becoming resistant, and there's lots of scenarios that we've seen. Gram-positive cocci, like staph, frequently found in wound infections of the skin and other places. In the 70s, most of the strains that were out there could be treated with drugs that are derivatives of penicillin. But you already know this. Already by 2004, 60% of those guys were resistant to this one type of penicillin-related derivative called methicillin. So we get this MRSA, or methicillin-resistant Staph aureus. And I think I mentioned to you already, if we took throat swabs from this class, there are about 25 of us in here. If we took throat swabs right now, about 8 of us would have Staph aureus in our throat. And about from this class, maybe 1 or 2 of us would have MRSA down inside of our throats right now. Because MRSA can get into normal individuals, and some people are just carriers. So can staph colonize the throat. That's typically where it likes to get, throat or nasal passages. It'll colonize there and typically not cause disease for those people that are carriers. Their body's acclimated, adjusted to the presence of that bacteria, and the bacteria lives in a kind of homeostasis with the body and doesn't cause disease unless something gets kicked out. But those guys can be spread to other people, and that's one of the problems. The other side of the problem is that we have about 30% of us have staph in our body. We take an antibiotic like a penicillin derivative, we're actually selecting for those guys that the antibiotic doesn't hit. We're selecting for more resistance in those strains. And so this next phenomena that's down at the bottom here has become a much bigger picture, healthcare associated MRSA infections. And that's really when things got brought to the forefront. Basically, it was those organizations that Obamacare is trying to fight against. All the healthcare agencies said, wait a minute, this patient went into your hospital for this procedure, and they came out with a staph infection, or they came out with a C. diff infection, or they came out with some other infection that they didn't have when they went into your healthcare facility. And the insurance companies were saying, that's not our responsibility to treat that patient. They got sick when they were under your care. And so it's the healthcare provider's responsibility to treat that illness that they got when they were in the hospital situation. And the healthcare providers are saying, wait a minute, that's a lot of money. We're going to go bankrupt if we have to deal with that. So it should be a good thing, right? Both sides are wanting to limit these healthcare-associated diseases, and because nobody wants to pay for them. And so that's what brought it to the forefront, was both sides were saying, well, wait a minute, we don't have a way to control. So most of the healthcare providers, hospitals now, when you go in for an inpatient treatment, before they admit you to the hospital, one of the first things they're going to do is swab your nasal passages. They're going to take that swab, they're going to go back, and they're going to put it on a plate that selects specifically for Staph aureus to see if you're already colonized with Staph aureus. Because if you get a surgery at some other site and you have Staph aureus and you get busy and accidentally transmit it to that wound site, then the healthcare provider wants to be able to say, hey, it's not our responsibility. Your patient already was infected, or at least carrying that particular strain of Staph aureus when they came to the hospital. They didn't get it from the environment. They got it from themselves. And that way they can limit a lot of the so-called healthcare-associated infection processes that happen. And so if you ever get admitted to Scott and White, they started doing this about 4 or 5 years ago. They'll swab your nose, they'll culture it, they'll save it, and they usually save it for several years just in case the insurance company comes back later and says, hey, my patient got this while they were under your care. They can just go back and say, look, it's the same strain that they had when they were here, when they first came into the hospital. It's not one that they picked up in the hospital setting. So we still have vancomycin. You guys played with that in the lab the other day. It's one of our last resort drugs. That's why a typical course of vancomycin costs about $900 here in the U.S. if you don't have insurance. Doesn't cost nearly that much to make it, but I think that the industry has wanted to say, doctors, you guys prescribe way too many antibiotics. You don't need to prescribe that many, and this is one of the last ones that works against some of these strains. So we're going to make it difficult for you to prescribe it, and not everybody can prescribe it. When I lived in China, you could walk into the pharmacy, you could say, oh, I've got a sore throat. The pharmacist would say, here, take this. They'd give you an antibiotic. Or you could go in and say, I'd like 3 courses of vancomycin, and they'd hand it to you over the counter without a prescription. And the U.S. is trying to prevent that type of situation so that vancomycin is not just thrown at everything, because vancomycin has some problems too. If you go to the microlab, there's a poster I did with people at Texas A&M Health Science Center and Scott and Wyatt on some research looking at Clostridium difficile infections. And vancomycin is one of the drugs that's thrown at patients that have C. diff, and it works well against C. diff because it works well against gram-positives. The problem is in the gut, besides C. diff that lives there normally, there's other organisms that are resistant to vancomycin, and those guys, many of them are cocci, so we call them enterococci, and they're the vancomycin-resistant enterococci. And if you throw vancomycin at the C. diff, you wipe out a lot of the bacteria in your gut, and then those resistant enterococci grow up. And the study that we did, part of it's published there, shows that those people that suffer from C. diff that get treated with vancomycin actually have a much worse prognosis, because the vancomycin-resistant enterococcus is even more pathogenic than the C. diff, and they usually can succumb to the vancomycin-resistant enterococcus. So lots of side effects of taking antibiotics in the process. Did I see a hand, Sarah? Did you?
I was going to say, I see about 1 or 2 VREs.
Yeah. We'll talk about how those guys came into existence. There's a couple of different pathways, but that poster also discusses some of them are just naturally resistant. They have a low-level resistance, but some of the others got higher-level resistance through some of the things that we're going to talk about in this chapter. So organisms, we've already said, need to adapt to their environment. And we've talked about in the previous chapter how natural selection helps them adapt. And that's what we do when we throw antibiotics at a global population. And anytime you take a systemic antibiotic, orally or IV, every bacterium in your system is getting hit with that antibiotic. Those that are a little bit resistant, you select for those that are even more resistant when you throw that antibiotic, so that you're selecting for those that have greater fitness. And bacteria know they need to be able to adjust to those new circumstances. They can do that through gene regulation that we just talked about, and sometimes they do it through the process we're about to talk about, which is called genetic change, or in other words, mutations that arise in their system. And so bacteria, you already know, are excellent genetic models. We can study them and look at how genetics works. One of the reasons is we can grow lots of them. The other reason is that we can get large numbers of them, and also we can manipulate them very easily genetically. We can throw genes in and out and say, make this protein, don't make this protein. We can turn things in and on and off in the laboratory. And so we can alter the genotype or the genetics of a given organism by tweaking their genome and throwing in new genes or killing different genes that are down inside. And then it's very easy to sequence the chromosome of bacteria now. And the other thing that's important, that why bacteria are, we see lots of change in bacteria very quickly, is because bacteria are what we call haploid. They only have 1 copy of each gene, because they typically only have 1 chromosome. In your cells, you've got 2 copies of each gene, at least, unless there are multiple copies on a chromosome. You've got at least 2 copies. You're diploid. So you get a mutation in one of them, and now one of them is non-functional. I have to make sure which finger I bend down. And then you still have 1 functional copy that can provide that protein that is needed for that particular function. And so you've got some protection against mutations, because even if that one goes defective, the other copy hopefully is still working. Now if you get a mutation in that one, now you don't make that protein, then things start to show up. But if we want to make mutations in bacteria, it's much easier, because we only have to hit the 1 gene. We don't have to hit the same gene on both copies of a chromosome, because again, they only have 1 chromosome. And so it makes it a lot easier to select for mutations. And when we get those changes in the genome, that makes a genotypic change. We say it changed the genotype. So when we talk about strains, strains of E. coli or strains of influenza or strains of any other organism, we're saying there are genetic differences in the chromosome between those different organisms. I just told you about E. coli 0157 picking up a big chunk of a chromosome from its cousin. It actually picked up nearly a quarter, nearly 25% the size of its original chromosome. Normally it's about 4 million base pairs, 4.5 million base pairs for an E. coli chromosome. It picked up a whole million base pairs from its cousin, brought it in, stuck it in its chromosome, so it got a lot of extra genetic information. But there's all kinds of, if you just make one nucleotide change different in a promoter. Remember in the lab I told you about those biovars? That's an easy biovar. If you hit the promoter and the promoter no longer works, or you hit one of the codons and it makes a stop codon in the middle of that open reading frame, just one nucleotide changes the genotype of that particular organism. And let's say you got that mutation down in the middle of the lactose gene, one of the genes that's needed for utilization of lactose in E. coli. Normally E. coli is going to use lactose. You've seen that before in the lab. You're going to see it in some of your unknowns. It should use lactose, but if you get one mutation, one nucleotide change down in the middle of that operon, it could kill one of those enzymes, and then it won't be able to use that lactose anymore. And that's a change in phenotype. Phenotype is what we can observe. So when you're recording culture characteristics in broth, or culture characteristics on plates, or all those biochemical properties, those are phenotypes. Those are things that you can observe. But each one of those is dictated by what's going on at the genome. So genotype influences the phenotype. That one strain of E. coli got much more pathogenic, can easily kill us if there's enough of it, just because of that change in the genotype. It's not the same E. coli anymore. A lot of people are debating, do we call all of these E. coli? Some of them are almost as similar to these other guys as they are to E. coli. What do we call them? And that's because of big changes in the genotype, and those changes in the genes can easily influence the change in the phenotype. There are some times when changes in the genotype don't influence phenotype. If you get a mutation in the chromosome in the middle of an open reading frame and it changes the codon from one of the codons to one of the other redundant codons, that won't change the phenotype as long as it can use that codon, but it's still a mutation. In that case, we have a change in the genotype, but we don't have any change in the phenotype. So hopefully you're starting to get an idea how the two can relate together. We'll talk about this a lot more as we go through. So in bacteria and in all organisms, we say that mutations happen. We know that, and we can't deny that mutations happen. And so now we're going to say that this cell divides into two. One goes down here, one goes here. This guy has a mutation that happened, a spontaneous mutation. Just to see what you remember from Chapter 7, how could spontaneous mutations arise? Probably not necessarily due to the environment. If there's some environmental influence, that's probably going to be an induced mutation.
On the DNA polymerase, it can transpose.
Remember DNA polymerase, it's got that little problem that we call error prone. And when it is error prone, it introduces changes, and as it's replicating the strand, it might put the wrong base in. And if that strand that's got the wrong base, if it's replicated again before that's fixed, then you got a mutation. And that's what we call these spontaneous mutations. The point of this figure is saying if this cell gets spontaneous mutation somewhere, all of its progeny are going to get that mutation. Okay, so that's basically how mutation happens. And over here in this bottom picture, this guy has a plasmid, but it transfers the plasmid over to this cell, and then all of the progeny of that cell now also get that plasmid. And so this process is called horizontal gene transfer because that initial transfer is coming from one cell to another this way that wasn't the progeny. And this would be vertical transfer because of the way it's flowing down through. So mutation goes be a vertical transfer, and then down, and this starts by horizontal transfer, but then you get vertical transfer on down through the population. Okay? That's really all the stuff I have time to tell you today, which is kind of sad, but we'll see.